Hi everyone, I am Guanghui Lin. You can call me Veronica. Here is my email. And my lab, the name of my lab is microbial genetics. I'm Guanghui Lin from Tzu Chi University. I'm the professor of School of Medicine, Department of Microbiology and Immunology. And I'm also the Dean of International College. So in the following, I'm going to introduce what I'm doing in my lab and what is my research work. So the detailed article, you can visit our website to see what kind of publication we have. Since our name is microbial genetics, so we are working some micro, especially bacterial. We have studied some new species and which was isolated from hot spring of Taiwan. So we give it the species name as Taiwanensis, such as Myosomus taiwanensis. So we also work on Bacillus subtilis, Thermus thermophilus, Vibrio parahemolytics, and Rhodococcus jatis. Nowadays, we are focused on the study of Acinetobacter baumannii about the gene regulation. And during the SARS-CoV-2 pandemic, we also have one publication about SARS-CoV-2. So with people hanging out around with bacteria, we are the people working with bacteria. And who did we play with? We studied about alcoholic bacteria, which is bacterial Acinetobacter baumannii. It can degrade alcohol. So the main topic of the work, if you are interested in, is we are working on a bacteria which can degrade alcohol and how is the gene regulation. We also work on one bacteria which is bacterial degrading aromatic compound, which is Rhodococcus jatis. In the meantime, we also work bacteria from hot spring. Yes, you can find some new species which are also isolated from hot spring, which is Thermus thermophilus. So what kind of question we are asked? We are asking about the question of metabolism gene regulation of IAA, indole, and ethanol. So far, we have focused on ethanol degradation and how is the gene regulation in Acinetobacter baumannii. And we also work on biofuel production of Rhodococcus jatis. In the meantime, we also work with Thermus species. We are studying of how the bioinformation and quorum sensing. What kind of method we use? We are studied by biochemical function, genetic function, as well as gene regulation. So in the biochemical function, we have the basic technique about gene cloning, protein expression, and we also equipped with the technique for mutant construction, and also protein fusion and RNA expression analyzing by quantitative RT-PCR. And how did we study the gene regulation? We use proteomic study as well as transcriptome study. Here is the bioinformation analyzed under the light microscope and the electron microscope. We also express the protein and purify the protein. So how do we express the protein and purify the protein? The things we can use. In the following study, I will give you a brief introduction about the SARS-CoV-2, how do we use this technique to construct a diagnosis kit. For study the gene regulation, we also use the technique of EMSA. So we are working on Acinetobacter baumannii, and it is a gram-negative bacterial, and it is highly antibiotic resistance, and it also causes nosocomial infections. Also, it was found in the rhizosphere of plant, and it is IAA degradation and production bacterial. And we also know that it can metabolize ethanol. So here is the electron microscope of the Acinetobacter baumannii, and we also put some fluorescence gene into the bacteria to see how the gene expression being regulated. So here is the structure of IAA. So what else? We use G. megalena as the animal model to study how the variance of the wild type and any kind of mutation strain. So here's the structures. And nowadays we are studying on two component system which composed of sensor and regulator, and how is the two component system regulate IAA metabolism, alcohol metabolism, and memory stress resistant, as well as alcohol, the other kind of alcohol metabolism. So in the above, we introduce how did bacterial gene regulation. While we are using those techniques for gene cloning, and how do we apply for the SARS-CoV-2 study? So here's the model of a coronavirus, and the protein of the nucleocapsid protein is the protein for diagnosis, and M protein is the highly expressed protein in this virus, and S protein, spike protein, is the protein for vaccine. And here's the structure of the spike protein. But today I'm not going to introduce it in detail. While we are using to produce the SARS-CoV-2 antibody detection kit, we will need a purified M protein. So we use E. coli as the model to express M protein. And here's the publication we have for a coded optimized SARS-CoV-2 M protein, which we produce M protein to generate the protein to construct the diagnosis kit. So while we get the E. coli clone, and we grow a mass amount of bacteria and disrupt the bacteria, purify the protein in the cold room, as well as in the by the machine, which with size exclusion chromatograph column. And while we get the purified protein, and we can construct the diagnosis kit to detection. Here is the brain we produce the antibody diagnosis kit for SARS-CoV-2. And we also have a patent strain for the M protein production. So except the academic part, what do we do in our laboratory? We have some regular academic activity. We have weekly academic laboratory meeting. In the lab meeting, you have to report some academic article. Maybe a semester you have only report one article, and you also have a progress report monthly and a weekly report, as well as you can discuss your progress with me in person per week. So during the summer, what do? We will attend the Conference of Bacteriology. It was hosted in Taiwan, and last year we hosted the conference in Tsinghua University. And next year, 2023, we will attend the FEMS Congress in German, Hamburg. So if you have fruitful data while you join my lab, you can submit your research data, article, and abstract to the FEMS Congress. So if your abstract being accepted, you might get the financial support from me and to attend the conference. Maybe part of the data is good enough, then you can join the conference, and I can give you some part of the financial support to attend the conference. So we are saving money now. Okay, here's some pictures. We also have a photo with the Dean of Academic Syndicat, which is the most big research institute in Taiwan. And here's me. So while we are attending a bacterial conference, and we have a group meeting with each other. The T-shirt is from a student from SRM who designed this T-shirt. Okay, so what we have to do, except academic activity, we have some happy activities, such as we have meal together every semester or annually or occasionally. We have some special activities such as maybe someone's birthdays or what else. And during the summer, we have the lab trip where we are attending conference of bacteriology. So next week, we are going to have the lab trip in Taipei and Yilan. So here are some of the happy photos together. Okay, so you are very welcome to join Microbial Genetic Lab. You can feel free to contact with me with the email, and you can visit our website in the following to see what is our publication. So welcome all students from each school. You are very welcome to join my lab. Thank you.